Okay, so I'm going to talk about a long-ranging dispute about human evolution. And it makes me question some of Kuhn's ideas about paradigm shift, where he talks about one paradigm and then another paradigm replaces it, kind of the mono-paradigm hypothesis. And also I'm going to talk about the idea of falsification of null hypothesis, which was advocated by Karl Popper. And I think these two things are very interrelated with respect to this debate. So what is the debate that I'm going to talk about? Well, first of all, we've already talked about Popper and the idea of hypothesis testing. And Popper had the idea that if you have a null hypothesis and you can reject it, that's actually the strongest inference in science. Accepting the null hypothesis because you did not reject it is a weaker form of inference. So basically he's saying science is better at knowing what is false than knowing what is absolutely true. And I think that certainly is the case. So is this, and Popper has argued that this kind of backwards way of saying, okay, I'm trying to reject a null hypothesis is rather unique to science, and he thought it was one of the strongest inference methods in science. And Kuhn also argued that even distinguished scientists often ignore data or observations that are incompatible with the favored hypothesis. But I'm going to try to give you an example where I think very intelligent, very informed people still can end up having very deviant views about an issue. So what I'm going to talk about is a very old argument, and of course it goes back to Darwin. Okay. So if you look at the origin of species, that has one figure in it, okay, and that figure is this evolutionary tree. Let's see. Yeah, so Darwin presented this. It's a hypothetical tree, so he has current species at this line, and he's showing that they can have a common ancestor, but many lineages in the past had gone extinct. And so this was the idea of an evolutionary tree, and it was very, very popular with many people, including Darwin. But even though he actually didn't give an actual evolutionary tree in origin, he did have this hypothetical tree. When he wrote his next paper, his next book that dealt with evolution, The Descent of Man, there are no evolutionary trees in this book at all. None. But it's not because he didn't think of evolutionary trees there. For example, in his notebooks, we have an evolutionary tree of primates that shows man as part of it. But Darwin deliberately did not want to put this into his book because although he had this idea, he just felt that there wasn't really enough information at the time to really have a justifiable evolutionary tree. It was too speculative. However, many of Darwin's supporters, initial supporters, were not inhibited at all about creating evolutionary trees with very little data. Okay? And one of them was Ernst Haeckel. Most of you probably know him from the quote, "Ontogeny recapitulates phylogeny." Okay? So he was a very early supporter of Darwin, and he loved evolutionary trees. And so as early as 1868, he had an evolutionary tree, not just of primates, but of human populations. And he had two major branches in humans. And in each subsequent decade, he kept increasing the number. So by the end of the 19th century, he had this tree of humans. These were all human, contemporary human populations. And he showed 12 distinct lineages of humans that were all on separate branches. Okay, very distinct, and in fact he thought they were so distinct he called them species, so that there are 12 contemporary species of humans. Okay. Now Darwin didn't think much of this. We know this from some of the correspondence Darwin had with Haeckel, and Darwin was heavily influenced by his experiences on the Beagle. He got to see some of the human diversity in a way that very few people at that age got to see, including Haeckel. But moreover, he spent a lot of time in Brazil. In fact, he spent more time in Brazil than he did in the Galapagos. And this time in Brazil was very important for him because he quickly learned that the concepts of race that Englishmen had did not correspond at all to the concepts of race of other people. Moreover, he saw hybridization going on rampantly in Brazil, and the people were perfectly normal people. They weren't degenerate in any way. And consequently, he wrote in The Descent of Man, quote, "But the most weighty of all the arguments against treating the races of man as distinct species," this was focused at Haeckel, "is that they graduate into each other independently in many cases, as far as we can judge, by their having intercrossed." So he did not see distinct branches of humans like Haeckel did. He saw a continuum, and he saw lots of interbreeding, what we would now call gene flow, rather than just lines of descent. So he argued very strongly against any evolutionary tree within humans. And Haeckel, of course, argued just the opposite. Okay, so Hegel was wrong to treat races as separate branches. Darwin even questioned whether there were races at all because there was a continuum. Well, this debate has never gone away. Okay, even though our technologies have changed in the last, 160 years, our analytical abilities have changed, this dichotomy about the role of gene flow versus population trees in humans has persisted almost identically throughout this entire time period. So, and we're going fast forward now to the second half of the 20th century, and if you read the literature at that time, you'll see that there were three major models of human evolution that were presented. One was this out of Africa hypothesis here. All of them agreed that the human lineage originated in Africa by this time. In the earlier part of the 20th century, even that wasn't a consensus. But now the fossil record was clear, humans evolved in Africa. And originally they thought about 1 million years, now we know it's much closer to 2 million years. Homo erectus came out of Africa and colonized Eurasia, at least part of Eurasia. And so in the out of Africa model, they formed separate lineages. So there was a tree, an evolutionary tree again. And this tree went on, but finally in the out of Africa replacement model, sometime around here, you started having the evolution of many anatomically modern traits. Primarily, but not exclusively, but although it's often presented as Homo sapiens suddenly evolved just in Africa. And that's an oversimplification, but I don't have time to talk about that. But anyway, humans evolved here, and then these anatomically modern humans came out, recolonized Eurasia, and drove these earlier Eurasian lineages to complete extinction. And they were completely replaced. And now you had another tree of these populations. So in this model, there's once again no role for gene flow. There isn't any gene flow in this model. Even another model advocated, particularly by Carlton Kuhn, was the candelabra model. Starts out the same, but there was no secondary expansion of humans out of Africa. Rather, he argued that all the humans, like Haeckel, evolved independently from their common origin of almost 2 million years ago into their modern forms. So there was massive parallel evolution in the candelabra model. But then, starting in the 30s and particularly in the 1940s, a German anthropologist by the name of Weidenreich proclaimed or proposed the multi-regional model. And he said, yes, humans came out of Africa, but then these lines, these diagonal lines, are all indicating gene flow. And the gene flow was mostly between adjacent populations, but he said there were really no barriers. So a gene could use a stepping stone model, which Wright came up with, that would allow genes to spread throughout all of it. So after this point, there is no evolutionary tree at all. There's a network, a trellis that goes on. So even though with isolation by distance you can have local genetic differentiation, it still evolved as a single lineage. And this was quite controversial at the time. And interestingly, Dojansky, who you heard about earlier, who did not work on humans, wrote a paper in the American Anthropologist saying, this solves all the problems. Okay, we have all of humans evolving as a single lineage with some local differentiation. And gene flow is playing the very most important role in this evolution of modern humans. Now, things got really stirred up in 1987 when Rebecca Kahn et al. Out of Alan Wilson's lab published this paper on the mitochondrial DNA haplotype tree. So here's yet another kind of evolutionary tree that should not be confused with a species tree or a population tree. This is a tree of just one piece of DNA. And if you have a piece of DNA that hasn't had much recombination or no recombination, like mitochondrial DNA, you can reconstruct the evolutionary history of all the variation, the haplotypes that exist today. And you can construct an evolutionary tree of how they coalesce back to a single common DNA. And I want to emphasize, this is only the evolutionary history of that piece of DNA. It's not necessarily a history of the populations at all. And we'll give an example of that in just a second. But anyway, so this was their haplotype tree of mitochondrial DNA in humans. And they said we have one clade or branch that's found only in Africa, and another major clade that's found all over the world. It's found in Africa, but also Eurasia and other places. And the ancestor was rooted into Africa. So they said, and moreover, they estimated the age of this common ancestor at only about 200,000 to 300,000 years. And so they said, well, you can't be the candelabra model, because the candelabra model says all of humanity only had a common ancestor almost 2 million years ago. So they could say we falsified the candelabra model. But they went beyond that. They said, well, the candelabra model is exactly the same as the multi-regional model. In fact, they only called it the multi-regional model. And here's the direct quotes, because people don't believe this. Okay, an alternative. Notice it's a single alternative. An alternative. An alternative view of human evolution rests on evidence that Homo has been present in Asia as well as in Africa for at least 1 million years. Now we would say it's closer to 2 million. And holds that the transformation of archaic to anatomically modern humans occurred in parallel. That's Kuhn's model. In parallel in different parts of the old world. And they give two references, 33 and 49. 33 is Wolpoff, who was the modern advocate of the multi-regional hypothesis of Weidenreich, that put gene flow as being very important, saying we had local differentiation due to local adaptation and isolation by distance, but all of humanity had been evolving as a single lineage. Kuhn, of course, was the one who advocated the candelabra model. You couldn't ask for two models that were more different than Kuhn's complete tree with no role for gene flow versus Weidenreich that says there's no tree, but there's a lot of gene flow. They were total opposite models, even though Kahn equated the two as a single alternative. And we're still dealing with the confusion of that. She called it the multi-regional model, but she was really talking about the Kuhn model that she falsified. And there's still confusion today about what the multi-regional model actually represents because of this. So she said, okay, we've eliminated both of these models because they're the same model. But are they? No, they're not equal at all. They're the most different models of human evolution you could come up with. And they equated them and said that we falsified multi-regional evolution. But that's not true, because under multi-regional evolution, the common ancestor of the mitochondrial DNA could be in any location where humans lived at that time. Moreover, estimates of the size indicated the bulk of humanity at that time period was found in Africa. So under standard coalescent theory, which is kind of the backwards population genetics, the most likely place for the origin of the mitochondrial tree is in Africa under the multi-regional model. So in fact, this tree is completely compatible with both out-of-Africa replacement and the multi-regional model. So what you've really done is they've only eliminated the candelabra model because it's not the same as the multi-regional model, even though they misstated that and caused tremendous confusion. Now I'm going to go forward again and get to some of my work. One of the problems with overlaying these haplotype trees on geography is there was no statistics, no hypothesis testing. People would just overlay it and then make up stories about it. And so we went from the field of phylogeography to statistical phylogeography. And so I came up with a method called nested clade analysis. There's no time to go through the details. All this is published though. But anyway, it uses the haplotype trees and converts them into a nested design. It does not assume the haplotype tree is a tree of populations or a tree of species. It's just the tree of that piece of DNA. But coalescent theory still says there's information about population level processes in that tree. But it's not just a simple overlaying a topology on one and equating that topology of a haplotype tree to a population tree. And moreover, the first thing you test is whether or not you've actually sampled enough that there's actually a real geographical association between the haplotypes you have and the geography that you're looking at. And that was never done before. So that was the first step, and then you interpreted those associations with coalescent theory. We applied it not just to one area of DNA. We did it with mitochondrial DNA, Y-DNA, and several nuclear regions that showed no recombination. And then you could cross-validate, and we came up with a formal maximum likelihood, log likelihood test mechanism for testing hypotheses. So it's a very different way of going about it. And with nested clade analysis, you have no prior models of human evolution. It's unlike these other cases where people are trying to show their model is right. We're just saying, here's the data from all these haplotype trees, and we let hypothesis testing determine what the model is, with no prior information at all. So the first thing nested clade analysis indicated was an expansion at the Homo erectus stage that was dated to 1.9 million years. And so that's consistent with all the things that you saw in the previous slide. In this area, right after that occurred, we could not reject the null hypothesis of no gene flow. That is, a population tree was the null hypothesis, and we couldn't reject it in this early Pleistocene time period. And so notice we got P levels on everything. So we're really doing Popperian hypothesis testing. This is the only part of this tree that's based on the acceptance of a null hypothesis. Everything else is based on a rejection of the null hypothesis, the strong inference under Popperian hypothesis testing. And the next thing we see is an expansion out of Africa, but this is not the one that was the controversy about. This was occurred in the mid-Pleistocene, and I don't have time to go through all of it, but it's strongly supported. It corresponds to the movement of the Acheulean II culture, which originated in Africa and then spread into Eurasia at this time period. It was also a very favorable climatic period for expansion out of Africa. It was one of these green Sahara phases, and in fact, many mammal species left Africa and went into Eurasia at this time. And there's a whole bunch of other evidence I won't talk about. But moreover, notice the lines aren't broken, because we also found out that there was significant admixture of these Acheulean humans who came out in the mid-Pleistocene with the earlier Eurasians. There was no replacement. Once again, more gene flow, more mixing. Then after that, we had significant gene flow for the rest of the time period. So after that, we had mostly isolation by distance, occasionally some dispersal, but long distance dispersal. But after that, all the human lineages in the old world were interconnected by gene flow under this model. But we did have another expansion out of Africa, the one that was always the controversy here, this one, where after many anatomically modern traits evolved, but they actually were found in more than one population. And this is the out of Africa one that everybody was worried about. But notice it also did not break the earlier lineages. Okay, and we rejected the null hypothesis of no admixture with the p of 10 to the minus 17th. Now, if you can reject a hypothesis of 10 to the minus 17th, that's about as, well, actually, I'll show you, it's not as good as you can get. So this was a very strong inference, okay? And this was all before ancient DNA, which we'll get into later. So here is what you get when you actually do formal hypothesis testing, okay? And notice we have a population tree in the early Pleistocene, but then we have a network of gene flow after the mid-Pleistocene on. So with hypothesis testing, you can have, if there is a tree, you can infer it. If there's not a tree, you can infer it. They're not, it's neutral to it. So when you're doing hypothesis testing, you can still have trees. Now, let's go forward even again. When I did this, I first published that in 2002 in Nature. I later got more DNA information, but the general outline really didn't change all that much as I added more gene regions. But other kinds of data came out that weren't haplotypes. So for example, there was a big study done on 57 different human populations, mostly with microsatellites and SNP variation, and this data set has been used by many people. One way that was done by Rosenberg et al., and I want to be careful, the way people have interpreted that and the way it was interpreted by Rosenberg was very different, okay? And so most of the conclusions that I'm against are not found in this paper, but they're based on this particular figure in the paper. Okay, they used a program called Structure, and Structure is a very popular program. It's used all the time. You can't go through any evolutionary paper now or book and not see structure analysis. And what it was done was, it was designed to say, if I specify the number of ancestral populations that existed, can I now classify people, put them into these number of bins that I've predetermined? And it does a very good job of doing that, okay? And so here by different colors you see, here's the Africans, here's the Europeans, the Middle East, here's the Asians, the Pacific Islanders, the Amerindians, okay? So it looks pretty good, and they're very distinct, as you can see. Now, of course, if you had two, you could say, okay, I'll only find two populations. What it did is it united Africa and Europe. And people didn't like that because that falsified the out of Africa replacement hypothesis, which was still the popular hypothesis at that time. Okay. They didn't like to go to K=6 because then the little Pakistani population appeared as one of the most distinct, quote, races of humans. So everybody decided to choose the 5 because that corresponded to most of people's racial concepts at the time. But there is absolutely no statistical test associated with what is the optimal number of populations. There's no statistics there. There's also no statistic to say, is this population different from that population? It doesn't exist. Are the sample sizes relevant? You can show that if you sample from a Hardy-Weinberg population, you can get sampling differences which will appear as structural differences in these things. As one paper put it, the title of the paper was Structure, Easy to Use, Easy to Abuse. There's no hypothesis testing in structure, but it gives you nice color diagrams and everybody loves them. Here's the same data plotted against distance, and it fits beautifully the isolation by distance model. You couldn't ask for a better fit. Okay, even my lizards don't fit quite as well as humans do. Okay, so anyway, and I've tried to show where it was. So they're colored differently, but that's the red from Africa and so on. And so here we have this model, and Do you see the distinct categories? Where are the distinct breaks that we see here, these absolute breaks? Where are they in the data? They're not there at all. This is an artifact of sampling, and people pointed that out very rapidly. It was just an artifact of the way structure was used. Now, another thing you can do is you can test for treeness. A tree is a particular kind of network that's highly constrained. But when you have gene flow, you have more complicated networks. So a tree is one that has only very few lines between the nodes, and it's very constrained, and it makes very predictions about the property of the network. And long ago, Cavelli, Sforza, and Piazza came up with a test in 1975 to test whether or not the underlying genetic data fits the constraints of an evolutionary tree. The trouble was, in 1975, the most populations they could study were 5, and that took them months to get the computer to do it. So nobody used their test after that, including Cavelli Sforza, because it just computationally wasn't feasible. Well, as we know, the technology's changed, and now it is more feasible to implement their test. And so Hundley wrote this paper, and they came up with this tree. Okay, and how did they get a tree? They calculate, well, they used a neighbor joining tree as their initial condition. Neighbor joining is an easy way to get a tree. You can always get a tree from any data set, regardless of whether the underlying data is tree-like or not. Neighbor joining will always give you a tree. That's what it was designed to do. And then they calculated the Cavelli, Sforza, Piazza test statistic, and then they started swapping branches and that, and searching the surface of this initial neighbor joining tree to find the tree that minimizes the test result. So they're trying to actually bias the tree that they end up with to accept the null hypothesis of a evolutionary population tree. So they use the Cavelli, Sforza, Piazza test statistic as an estimator of the tree. Interestingly, even though they did this, they never actually bothered to actually calculate the p-values associated with the test. So they used the test statistic as an estimator. And they said, this is the best tree of human populations anybody had, even better than Haeckel. Okay. Now, the trouble is, since they didn't do it, I decided to do it. Okay. And here are the results. The total tree was rejected at a p-value of less than 10 to the minus 200th power. Okay, and that's because 10 to the minus 200th was the lowest value my computer would allow. Now, is this a tree-like structure? They said this is the best tree. They love this tree. And they said, oh, some of the subtrees are even better, and sure enough they are. If you just look at the African populations, you only rejected at 10 to the minus 110. There is no tree-like structure in the genetic data at all. Why? I don't know if they actually evaluated the test and just ignored it, but they calculated all the test statistics, but they never used it as a test. Okay. Now, it's based on the same data that we've already talked about. You notice one thing peculiar about this tree. All these are contemporary populations, and in an ideal tree, they would all have the same endpoint. That is, the branch lengths going back to the common ancestor should be the same. But here we have the Africans being very short branches, the Amerindians having very long branches. They go back to the common ancestor. Why is that?
Rooting artifact.
It's a rooting artifact. Okay, we're talking about the root is in Africa. The farther away you get from Africa, the greater the genetic distance, and therefore the greater these branch lengths. It's all an artifact because they totally ignored the role of gene flow and isolation by distance. The data fits this isolation by distance model very well. Here it's 10 to the minus 200th power, or below that. I don't know how far below. It doesn't make any difference at that point. Okay. Now, how about the ancient DNA? So this is another technological innovation. We now get another way of getting at the past. So all these studies were done on contemporary populations and contemporary surveys and reconstructing the past, but now we have a way of actually getting genomes from some fossils. We don't have a lot of them, but we do have enough that we can do some things with it. Okay, what the fossils clearly showed was that in fact there was not replacement. Okay, as you probably know, Neanderthal DNA, Denisovian DNA is found in modern humans. And people think it's not been very major because on the average it's been estimated somewhere between 2%, I've seen up to 4 or 5% of our DNA of individuals has these archaic Eurasian. But remember, we're really only talking about 4 high-quality genomes. And just when the Denisovan genome was added, suddenly the amount of admixture went up, way up. So these numbers can only go up. But also different people have different ones. So how much of the Neanderthal and Denisovian genome is found in the modern human gene pool? Anyone want to take a guess? Yeah, 50% of these archaic genomes have been absorbed into the modern human gene pool. So it's not like Neanderthals and Denisovans went extinct, they were just absorbed into the modern human gene pool. So that's certainly said, okay, the out-of-after replacement hypothesis, everybody now accepted it wasn't a complete replacement, there was admixture. And, but now let's go back to this idea of testing a tree. Reich et al. In 2009 came up with a way of testing whether or not the archaic DNA was indicating there was a tree in the past, or there was a tree leading to the modern lines of humans. And there's a whole series of tests that are all interrelated. I'm not going to go through all of them, but what I'm saying actually is applicable to all of them. Remember, we don't have many genomes, so they just did a 4 taxa test. In some cases they used a 3 taxa test, some 5, but always a very small number. So they assume you have a tree. You've got 4 populations or 4 genomes, and generally speaking, P4 is an outgroup. Sometimes it's a chimpanzee, sometimes it's an archaic human. P3 is also an old human, and P1 and P2 are often, say, contemporary populations. So this top tree here, this is the tree that they say we picked the populations on purpose, that this is the tree that represents that. Okay? So that's the first thing they did. So then they say there's no gene flow, so the null hypothesis is that tree is a true population tree. Okay? And then they say that let's let capital A be an ancestral allele. That's the allele they find in the outgroup. And let there be a mutation that left to a derived allele, which we'll call B. And moreover, we will only look as we scan the genome, those nucleotides in which there's exactly 2 B alleles and 2 A alleles. So ideally, under that tree, the B allele was a mutation on that dashed line. So that would result in the configuration, if you looked at this, going from P1, P2, P3, P4, you would have BBAA. Okay? But here's the complication. This is another reason why you should never equate a haplotype tree to even a species tree, much less a population tree. There's this phenomenon called lineage sorting. When a species splits, the initial populations share many of the genes, but the haplotype tree is older than the split. And you can easily have, just by random genetic drift, you can have, say, an allele here that actually traces back to this other species, the wrong species. This is called a transspecific polymorphism. And we have them. In fact, in humans, we've even found a few that go all the way back to rhesus monkey. Okay, really, so and haplotype trees are not species trees or population trees. Or sometimes you'll have this. So like here, once again, here's an allele that goes back to species B versus the other alleles in the same species that go way back there. Okay, you can have these, lineage sorting will cause these topological disharmonies. And do they occur? Well, here is one of the original examples. They looked at 23,000 gene regions in the nuclear genome for humans and the great apes, as well as the rhesus monkey as an outgroup. And then they found about 11,000 of these trees were statistically very accurately estimated. And so they confined their estimate to these reliable haplotype trees. And what you see is that about 79% of these reliable haplotype trees said that humans and chimps were the sister species. And with a lot of other data, this is the accepted species tree for these primates. However, notice that we also have about 10% of the trees saying that gorillas and chimps are the sister species, and another 10% say that humans and gorillas are the sister species. These are not mistakes. These are the true haplotype trees. But because of lineage sorting, the topology of the haplotype tree is discordant with the topology of the species tree. And when you get down to population trees within a species, this problem of lineage sorting increases by magnitude. So when they were doing these tests here, the Abba-Baba test, why is it called Abba-Baba? Well, the BBAA is, quote, the true tree, but they know due to lineage sorting, we expect to see lots of discrepancies. And so it's the discrepancies that this test focuses on. So we do this, we condition that all everything is compatible with the tree because there was no gene flow, but the incompatibilities arise due to lineage sorting, which is expected under coalescent theory. And there's two types of incompatibility. So for example, in this, if you use this as your tree, it's really saying 3 has the A allele, but it should have the B allele, but P2 now has the B allele. So this creates the order like this. This one is, P2. So this is A, B, B, A. This one is B, A, B, A. Abba-Baba. Everybody thinks it's pretty funny, but there's a whole class of these statistics. So we have ways of testing trees with archaic DNA, ancient DNA.
If you want to have a few questions, then...
Oh, okay.
Yeah.
Okay. Well, anyway, I'll just go on quickly. You can come up with a test that actually measures these two different deviations. If they're due to random lineage sorting, this statistic should be 0. If it's due to gene flow has interfered with it, you're going to get biases in this tree. And therefore, this is a tree for looking at it. And so you can test the null hypothesis of no gene flow. That is a population tree with the Abba-Baba test and these similar tests. Okay, and I'll just skip all of this since we're kind of going. Oh yeah, I should have said one thing. The problem is, how is the Abba-Baba test actually used in the literature? Remember, Hundley calculated the test statistic of Cavalli, Swartz, and Piazza, but used it to estimate a tree. The Abba-Baba test is done in the same way. When people get a significant value, what do? You reject a tree-like structure. That's what the test was designed for. But instead of doing that, they said, oh, because there's only really 3 taxa there, whenever I have a discrepancy, I can say, oh, there must have been gene flow between 3 and 2. And I can say one admixture event can explain the discrepancy. So I still say, I believe the tree is true, but I just have to add on a minimal number of interconnections between them. I'm minimizing the role of gene flow. And the trouble with this is, already by 2010, less than a year after the original test, it was shown to be statistically inconsistent as an estimator. What is statistical inconsistency? It means you're guaranteed to get the wrong answer if you have enough data. of all the properties of an estimator, and I've got a master's degree in mathematical statistics. If you say an estimator is inconsistent, you never use it, except in the area of human evolution. And so you see diagrams like this. Every one of these dashed arrows represents a significant rejection of the null hypothesis of an evolutionary tree. Yet you see a tree structure here. And with just all these little lines showing the single admixture events that arise from just using the simple Abba-Baba test and similar tests. Moreover, when you look at it more carefully and you look at other data, and here's just one paper, it really shows it was isolation by distance in multiple recurrent gene flow. This is all garbage. What every one of these dashed lines says is there's no tree. That's the only statistically justifiable conclusion you can make. And everybody's showing Wikipedia. Here's how Wikipedia shows human evolution. They don't even have the dashed lines. There's no gene flow in human evolution. Okay, so I'll just conclude by this. I've been very influenced by particularly this recent book that just came out by Wagner, not Günter Wagner, it's another one. And why do so many scientists ignore hypothesis testing? These people are not dumb. They're doing really cutting-edge work. Okay, and I think Kuhn's explanation about ignoring incompatibilities is wrong. He says many of these scientists feel that adding this admixture, that explains the difficulty. They have solved the problem. They have a coherent explanation for their data. Okay, it successfully resolves the incompatibilities. And Chang and Wagner, in their books and papers, argue that really what is happening is not one paradigm replacing another, but parallel paradigms exist because biological problems are so complex that different groups will approach them using very different methodologies in different ways. And they feel that they've explained things very well, but they're not co-measurable. So do you use these test statistics as tests, or do you use them as estimators? That's the divergence. And so even though we've had all this evolution of technology, of analytical procedures, computer power, both cases we have what the practitioners call a coherent theory of reality, in which coherence is the pragmatic ability of a system using very different non-commeasurable actions to carry out its intended task. And it results in a pragmatic understanding of truth. And that's what I think has occurred. That's why this two different parallel views of human evolution have persisted for over 160 years, despite all the differences in technologies that have emerged, because people are not talking the same language. And they don't recognize the validity of the other field. You can imagine which side I'm on. But as that Wikipedia showed you, the population tree paradigm, that's the one that dominates not only the popular literature, but even much of the scientific literature. I mean, you see those little diagrams with the little admixture lines all the time. It's pure garbage from a statistical point of view. But that's the way people think of human evolution.
Thank you very much. Can you post it? I got 2 calls already for the test, so.
Okay, very briefly. We learned from Ariel that we should no longer use the gene as a unit of calculation, but the network. But what you had there with trees, I mean, I'm sorry that we have to give up on the trees, but you showed it quite convincingly. But you used genes, and you didn't differentiate between regulatory genes or junk DNA. You don't believe in it, but I do. And or protein coding
there
genes.
is junk DNA. I just don't think it's all junk.
No. And protein coding genes. So how can these two be reconciled?
Yeah, we're really using only the genes and the haplotype trees associated with a particular DNA region. Whether it actually has a protein coding gene or not is irrelevant. the relevant thing for me is there's no recombination detectable in that region, so we construct a reliable haplotype tree. Now some of these could be due to selection, it could modify things. That's why we want to do multiple regions and then do a formal cross-validation, because we're trying to get the common signals that are due to historical events and demographic events and separate out the things that might influence a particular gene, say that's adaptive to high altitude or something like that. So that's why the cross-validation across genes is very important. You're trying to find the common signals that reflect the history of the population and not just the local properties associated with that one gene. So it's very dangerous to do phylogeography like just on mitochondrial DNA or just on any other one gene. You want that cross-validity. And now it's easier to get these trees than ever. There was a paper just came out earlier this year where they found 27,000 regions of the human nuclear genome where they can construct reliable haplotype trees. So this, you can really do huge statistics on this problem now.
Fascinating. Okay, thank you very much for this great session. And now dinner.
No, lunch.